Hello and welcome to our video series. In this episode, we are delving into experiences and insights of another remarkable member of our team. Uh, Hello, Peter. Please introduce yourself.
Hi, Kaloyan. Thanks for setting this up and a pleasure to talk to you and discuss about the past, future, and my vision about Scailyte. But first, to introduce myself to the audience. My name is Peter. I'm the CEO and one of the co-founders of Scailyte. Originally, I'm from Bulgaria and recently actually also received the Swiss citizenship. So this marks my origins, where I spent the first 20 years of my life. And now my last 15 years working and studying in Switzerland, now obtaining these two citizenships. And then from the education point of view, I studied biochemistry in Germany at the University of Tübingen. I also did a PhD in genetics at the University of Basel in Switzerland. And then later I worked as a sales specialist representing a company that is selling single cell and genomics tools. And this actually gave me the inspiration about Scailyte by seeing the potential of single cell technologies and single cell data and deciding that this is really the way to make a difference in biomedicine by actually unlocking the full potential of single cell data.
Thanks a lot for that. What inspired you to not join Scailyte? Because you're not joining Scailyte, you started Scailyte with the co-founders. And how has your role evolved since you started? So I guess maybe also on a day-to-day since the beginning of the company.
Mm. So in a few words, the inspiration came from the potential that I saw in single cell data for biomedical discovery, but in a little bit more detail, why single cell data? And this is maybe a question that I like to answer because I rarely hear people explaining what is the strength of single cell data over other types of omics data. There are now many different types of omics and biomedical data that are used for discovery. But for me, single cell data is really special in one way. And this is if you come to the basics of biology, how organisms are made and set up. Actually, the cell is the basic unit of life. So a living cell is made up of different molecules, and only as a cell, as a whole cell, it's a functional unit. So the molecules on their own, the DNA, the RNA, they're not functional as such in an organism. And then the cells, they make up the tissues and the organs, and they actually communicate with each other, and they're also responsible for picking up and responding to signals from the environment and from neighboring cells. That's why if you study the cell at the molecular level, and actually at high scale and a lot of throughput, then you can really get an insight in what's going on with an organism, what's going on in development and disease as the cells respond and develop according to certain signals. And this is why I see the single cell technologies being so powerful, because they study at the single cell level what's going on in an organism, what's going on in a certain tissue. And yeah, that's why I saw this huge potential of single cell data. And this gave me the inspiration to work on the single cell data and come up with discoveries that can be translated into clinical applications. And about Scailyte, so we're actually a data-driven company, not so much a tools company, because I see there already when I did my PhD, there were quite a few companies that have developed and commercialized tools for single-cell analysis. And actually the bigger bottleneck was the data interpretation, which is why with Scailyte, we focused really on this part, on the data interpretation.
Thanks a lot for that. Just as a point to continue into our quick discussion, can you share a recent project or accomplishment that you're particularly proud of?
Yes, one recent project that we did was in collaboration with our scientific advisor, Vijay. He's a professor at the Queen's University in Belfast and recently also moved to Denmark as a second chair there. And we partnered with one of his PhD students, Ryan Lusby, on a project where Ryan used SkyVision, our software, to interpret single cell data in combination with multiple sets of publicly available single cell and omics data to discover biomarkers that can predict brain metastases on a pan-cancer level, meaning that he took datasets from different cancer types, patients having different cancer, that all had brain metastases. And of course, as a control group, patients that didn't have brain metastases. And first, he applied SkyVision to actually discover the relevant cells and the relevant gene signatures. And then he used a number of other tools to validate this discovery, first also on bulk omics data, and to dive even deeper and translate this initial signature into a biomarker that could be then further developed as a clinical application. And for me, this is a true showcase of the power of our software and our discovery approach, and how it can also be used to extract more insights from already available public data, because this data has already been published, and now we made new discoveries from data that others have already analyzed with other tools.
And I guess perfect segue into the next question. What in your opinion, sets Scailyte apart from all organizations in the field?
Mm. Yeah, indeed. I already mentioned this in the example. I think one of the key strengths and unique features of Scailyte is the approach that we take to interpret single cell data, which is unbiased. So we do not put any prior knowledge when we do the discovery. And we let the algorithm discover which cells and which gene profiles, gene signatures are associating with a certain clinical endpoint. And only then we start with the interpretation and comparing reference gene signatures and so on. And I think this unbiased approach is really important in the beginning. And we also have an algorithm that is highly automated so we can get to an answer very quickly. So it's speed and discovery potential that sets us apart.
And just the final question, kind of to summarize it all and pinpoint a point in the future, how do you envision the impact of Scailyte into the future of healthcare and diagnostics?
So we're recently focusing on immuno-oncology, and we did also projects in various other disease areas, including also endometriosis. But I see the biggest potential impact that we can make is indeed in immuno-oncology. And this is the field where novel immunotherapies are developed for cancer patients. And this is a highly complex area also biologically. First, cancer is a very complex disease, and actually there are multiple diseases bundled together as or labeled together as cancer. And as cancer evolves, this change and actually how the patient responds to a certain treatment depends also, of course, on the stage of the disease, but also on what other diseases they have, on the environment, on their lifestyle. And this makes it highly complex. And also the treatments that act on the immune system, so these immunotherapies, they're also very complex and can trigger a very complex response in the organism. So coming back to my original answer on what I see as the biggest potential of single cell data, this is exactly where it can unleash the full potential of this technology, coming up with identifying what is going on biologically at the single cell level, understanding first what's happening in the body and as the disease progresses, but also how these new treatments modulate these cells. And that's where I think Scailyte can make the biggest impact by grasping this complexity and discovering biomarkers that can stratify the patients and identify patients that respond to a given treatment. So ultimately, finding the right patient for the right treatment in a very complex setting. And that's where I see Scailyte can and will make the biggest impact, and that's where we're headed now.
Thanks for that, Peter. And just to kind of leave the audience with a action or maybe just a statement from your side, what would you say to everyone watching?
Yeah, one statement I would say is collaborate and make an impact. And to explain it, what I see now also in immuno-oncology and generally in biomedical research, the field is becoming very complex and it's difficult for one player to address all the challenges and actually make a meaningful impact. It can only work in a collaboration mode. And that's also what we do at Scailyte. We collaborate with biotech companies, we collaborate with universities, and we leverage the strength of each partner. So that's my call to action or message to everyone. Be open and collaborate so that we can jointly make an impact in biomedicine.
Thanks, Peter, and thanks to everyone watching.
Thanks, Kalyan.